students. Today we are here to learn about the spoilage of the meat and the milk products. Why do we consume meat and milk? We consume it because meat and milk are very good source of protein. And the same holds true for the growth of microorganisms because they too want protein for their growth and for their survival. So let us learn few things about the spoilage of the meat and the milk products and what are the different types of microorganisms that grow in such substrates. If we consider the growth of microorganisms in meat, there are certain factors on which this growth depends on. The first thing we should consider is the physical property of the meat. Now, physical property refers to the shape or the morphology of the meat. Say for example, if I take a large piece of the meat and I take small pieces of the meat, or I can say the minced meat is more susceptible to the growth of microorganisms because there is a large amount of surface area and most surface area holds true for the growth of microorganisms. Therefore, the physical property of the meat very well decides the growth of microorganisms. The second most important factor is the chemical property. That means the composition of the meat. Now, meat is coming from different sources. It may be a poultry, that means a chicken. It can be a beef, it can be a goat. So, the composition of meat varies and therefore, the growth of microorganisms also vary. The third important factor is the availability of the oxygen. Whether the oxygen is there or not. Because of the presence of oxygen, it decides that which type of microorganisms are going to grow, whether they will be aerobes or they will be anaerobes. And the fourth most important factor that decides the growth on meat is the temperature. That means what is the temperature in which the meat is going to be exposed, whether it's a low temperature or a high temperature. Now, let us learn about the different kinds of the microbes, or we say the microbiota of the meat. If we go on to the spoilage of the meat, we find that numerous microorganisms are responsible for it. They may be bacteria, they may be fungus, or they may be yeast. Coming on to the bacterial cells, or the different species of bacteria that may spoil the meat can be the species of Pseudomonas, Acinetobacter, can be Morozella, Micrococcus, Staphylococcus or Shewanella, which gives a bad odor and a bad color to the meat. In addition to bacteria, we have many species of fungus which affects the meat and therefore cause the spoilage of the meat. For example, we have Thamnidium, Mucor and Rhizopus which produces whiskers on the beef meat. Also, Cladosporium is a fungus which produces black spots over the surface of the meat which can be clearly seen in this picture. Many species of Penicillium produces green patches over the surface of the meat and therefore this meat should not be consumed at all. Sporotrichum and Chrysosporium are another two fungus which produces the white spots over the surface of the meat and therefore this shows that the meat has been spoiled by molds or by the growth of the fungus. One thing should be taken into account that molds apparently do not grow on meats if the storage temperature is below -5 degrees Celsius. Therefore, if you are able to maintain the temperature, you can inhibit the spoilage that is being caused by a fungus. Coming on to the different species of yeast that can spoil the meat can be the different species of Candida, especially Candida lipolytica and Candida zellanoides. Also, the species of Rhodotorula are the responsible microorganisms which cause the spoilage of the meat. Now, after knowing the different species of the microorganism which are responsible to cause the spoilage of the meat, let us consider the spoilage indicators. How do you come to know that the meat has been spoiled? Because you are not able to see the microorganisms. That means you should have some analysis, or we say the biochemical parameters, which shows you that the meat has been spoiled. We call this the spoilage indicators of the meat. The metabolic byproducts which are being observed during this spoilage can be different type of diamines, especially cadaverine and putrescine. Now, meat and is a very good source of protein along with the free amino acids that is present inside it. Lysine is one such amino acid which is undergoing the process of decarboxylation, and as a result of this we have the production of diamine which is called cadaverine. On the other hand, ornithine and arginine are again two different amino acids which are subjected to decarboxylation, and we have the production of putrescine. Now these diamines are the spoilage indicators. So if you take a piece of meat and test for these diamines, and if you find the positive test for this, it shows that the meat has been spoiled. Now the question arises, how come the meat is being spoiled by the microorganisms, or how come the microorganisms are getting inside the tissues? Now a very common thing we should have in our mind is we humans are also infected, and therefore the notation says that to be a human you have to be infected. We have about 300 to 400 species of bacteria in the gut. The same thing holds true in the case of the different animals, and therefore we say that the invasion of different tissues is due to the load of microorganism that is present inside the gut of an animal. It shows that the intestine of the cow, the intestine of the goat, the intestine of the chicken, so from wherever source we are getting the meat, has been infected or has been populated by different type of microorganisms, and when the animal dies, these microorganisms try to invade the different tissues of the animal, and therefore when such a meat is consumed, it is populated with different type of microorganisms. So one of the factors which influence the invasion of the microorganism is the load in the gut of the animal. The second most important factor which decides the invasion of the microorganism in the meat is the physiological condition of the animal immediately before slaughter. This is very important because the meat we are going to consume is coming from an animal. So we have to see whether the animal is infected or not, whether the animal has been suffering from a disease or not. If we encounter such a case where the animal is suffering from a disease, we should not consume such meat because the microorganisms are already present in the body of that animal. So the physiological state of the animal is very important in order to know the spoilage of the meat. The third most important factor is the method of cleaning and bleeding. Now after the slaughter, we have to clean the meat, or the animal has been bleeded so that you get different types of meats. Now this method of bleeding and the method of cleaning is very important, and whether they are done it in a, or whether the slaughterhouses are doing it in a proper way or not. And the last most important factor which decides the invasion of microorganism in the meat is the rate of cooling. This refers to the rate of cooling of the meat. That means after the animal has been sacrificed, if the meat is allowed to remain in the room temperature for a longer time, then it may be encountering different microorganisms which may be coming off from the environment, which is not at all desirable for the safe consumption of the meat. So now we know the meat can be spoiled by microbes, the different type of microbes. Now let us examine, or let us learn that what are the different types of spoilage, or how simply by looking at the meat we come to know that the meat is spoiled. So we call it the types of spoilage of the meat. The spoilage of the meat can be taken in two different conditions. One is aerobic, that is in the presence of oxygen, and the other one is anaerobic, that is in the absence of oxygen. Let us study the spoilage which is taking place in aerobic condition, that is in the presence of oxygen. The most important change that we encounter in aerobic condition is the surface slime. This refers to the meat where we find that a slimy appearance is there on the surface of the meat, which is caused by the bacterial species of Pseudomonas, Acinetobacter, Morozella, Alcaligenus, Streptococcus, Leuconostoc, Bacillus and Micrococcus. So many bacterial species are there which can cause a surface slime. The change in the color of the meat pigment also takes place. We all know the color of the fresh meat. But if we come across a meat where there is a change in the color, that is a very good indication to know that the meat has been spoiled. You can have the shades of green, brown, grey, which is as a result of the production of the oxidizing compounds like peroxides, hydrogen sulfide gas. And because of all these, you have the change in the color of the meat, which is caused by the different species of lactobacillus, which is heterofermenter, and leuconostoc. Leuconostoc specially gives a green color to the meat. Also, we find the transitivity of the fat. That means the fat or the lipid that is present in the meat is being degraded with the help of the lipolytic bacteria. Why? Because these bacteria are the one which is causing the degradation of lipid, and therefore lipolytic bacteria are Pseudomonas and Achromobacter, and there are many species of yeast also. Phosphorescence is a very uncommon defect that is encountered, but this can be very well seen if you keep the meat in the dark and you are able to see a phosphorescence, that is a light coming out in that dark. This may be due to the luminescence created by a bacteria known as photobacterium. Various surface colors are also encountered due to growth of the pigmented bacteria. Say for example, we can have the red spot by Serratia marcensis, blue color by Pseudomonas cincinnea. We can have the yellow color of the meat because of the Micrococcus and Flavobacterium, the green-blue color given by Chromobacterium lividum, or the purple stamping ink color that is being given on the surface of the meat by many rods and coccus. The off odor and the off taste that is encountered in the spoilage of the meat is due to the production of the volatile acids such as the acetic acid, formic acid, butyric acid, propionic acid, or the earthy flavor that comes. That means the earthy flavor of the growth of the actinomycetes. So all these changes in the aerobic condition shows that the meat has been spoiled. Then what about the changes which is taking place in the anaerobic condition? That is when the oxygen is not there, totally in the absence of oxygen. Souring is such a change. That means you get a sour taste in the meat, which is due to the production of the organic acid like formic acid, acetic acid, propionic acid, butyric acid, or succinic acid. Also, putrefaction is such a change which is taking place in which we find that there is a degradation of the proteins. We are consuming meat because we want some useful protein from the meat. But if that protein is being degraded by the bacteria, obviously that meat is not safe to be consumed. And this putrefaction process can be carried out by the production of different compounds like hydrogen sulfide, mercaptans, indole, ammonia, amines, which is done by different species of Clostridium, Pseudomonas, and alkaligenes. The taint refers to the off color and the off odor, especially the bone taint which is being encountered in the spoilage of the meat, which refers to the putrefaction, that means the degradation of the meat next to the bone, and therefore this is called the bone taint. Coming on to the spoilage of the poultry, we refer to the meat of chickens. Now this meat can be spoiled by enzymatic degradation. That means the enzyme which is present in the meat which deteriorates and therefore gives a foul smell or odor. Also, the spoilage can be caused by bacteria. Especially, there are different types of bacteria which is present in the intestine of the chicken, which penetrates the different tissue and therefore cause the spoilage. The small amount of iron can be present in the water by which the chicken meat is washed. Now, this small amount can be 1 to 5 ppm, parts per million, which can cause the production of a fluorescent pigment which is called pyoverdine by different species of Pseudomonas. Now, all these refers to the types of spoilage that is encountered in a chicken meat. After meat, now let us learn about the spoilage of the milk and the different type of milk products. Now milk again is a very good source of protein, and not only protein, it has got carbohydrates and many other things, and therefore we call milk as an ideal medium. Whenever we consume a glass of milk, we say that it is good enough for our health. The same thing holds true for the bacteria because they also get everything in the milk. What are these? The milk has got a high water content, high amount of moisture, and therefore very suitable for the growth of microorganisms. The milk has got a neutral pH from 6.4 to 6.8 and therefore a very good pH for the optimum growth of the microbes. The oxidation reduction potential of the milk is about 0.3 volts and therefore this enables the aerobes to grow readily. The average composition of the milk, if I look into, contains about 3.2% of protein, 4.8% of carbohydrate, 3.9% of lipid and 0.9% minerals. If I go into the detail about the composition of the milk, we find that we have presence of fat, then casein, whey protein and minerals and salts. Lot many other things are there in the milk. Now besides casein and lactoalbumin, it has got presence of amino acids, which is again a very good source for nitrogen. The main carbohydrate that is present in the milk is lactose. Therefore, milk is a good substrate for those bacteria which are able to ferment lactose. However, those bacteria which are not able to ferment lactose do not spoil the milk because they are not able to grow in milk. Coming on to the microorganisms that are present in milk. Now, the microorganisms that are present in milk can be of two types. They can be spoilage bacteria or they can be pathogenic bacteria. Now, what is the difference between the two? The pathogenic bacteria are those bacteria which are capable of inducing the food poisoning. Therefore, pathogenic means they can cause the disease. This disease causing capacity is there because they have got presence of certain enzymes like peptidase, lipase, esterase, polymerase, beta-galactosidase. However, there is another group of bacteria which is called the spoilage bacteria. This refers to their capacity because they are able to hydrolyze or degrade the different components that is present inside the milk. Now, these components may be protein, fat, lactose, and therefore you have production of many byproducts which is not useful for the human and that becomes a harmful effect to the milk. The reactions or such reactions can lead to spoilage of the milk and therefore we have the off order of taste and the bad appearance of the milk as it can be well seen in this picture. Now, what about the composition of the microbe which is present in the milk? We have learned about the microorganism that is invading the tissue because they are coming from the intestine or because of the physiological states and many other factors. Now, what about the microorganism that is present in milk? Actually, the milk is produced from the mammary glands. Although we say that the milk which is produced in the mammary glands of healthy animal is initially sterile, but it can be infected because the microorganisms are able to pass inside the udder through teats. So, the milk which is drawn aseptically from the udder of an animal contain gram-positive coccus like Streptococcus, Agar-lacti, Staphylococcus and Micrococcus. We have presence of numerous lactic acid bacteria. Also, we can find the species of Pseudomonas, Corynebacterium and many species of yeast. Let us now learn about the spoilage of the milk and the cream. Now, temperature is very important factor as I told you for the spoilage. So, let us learn that how this temperature decides that which microorganisms are responsible for the spoilage. If the raw milk is maintained at a temperature of 10 to 37 degrees Celsius, then we find that souring occurs because of the growth of Streptococcus lactis along with the growth of coliform bacteria, enterococci, lactobacilli and micrococci. However, at a little high temperature at about 37 degree to 50 degrees Celsius, we find that there is a growth of Streptococcus thermophilus, Streptococcus faecalis, which produces about 1% acid, not so much. Rest of the acid is being produced by the Lactobacillus bulgaricus. So, all the microbiota together are giving an acidic pH to the milk if it is maintained at a temperature of 37 to 50 degrees Celsius. However, if you elevate the temperature a little bit at a temperature of 50 degrees Celsius or above 50 degrees Celsius, we find that the lactobacillus can grow, but it cannot produce the acid. The butyric acid is also seen to be produced in the milk by the action of the Clostridium species, and this inhibits the normal lactic acid formation. So because of all these, you can have the spoilage of the milk and therefore the cream also. Now the question says that if you have so many microorganisms in the milk which can spoil the milk, then why don't we go in for the pasteurization? Yes, we do go in for the pasteurization in order to make our milk more sterile, but we don't make it sterile. We rather get rid of the pathogenic bacteria so that they do not cause disease in the human body. But there are many bacteria which are still surviving in the pasteurized milk and they can cause the spoilage of the milk. Then what are those bacteria? These bacteria are the species like Pseudomonas fluorescens, Pseudomonas fragile, species of Bacillus, Clostridium, Corynebacterium, Arthobacter, Lactobacillus, Micrococcus and many species of Streptococcus. So all these are able to resist the high temperature that is given during pasteurization. The pathogens which are found in the milk or which can inhibit or which can offbeat the color and the taste of the dairy product can be these. Mycobacterium tuberculosis. Now this organism is responsible to cause tuberculosis and this is coming from the cow. Micrococcus pyogenes. This is the able microorganism which can cause the sore throat in man. This is again coming from the cow. Brucella abortus, which causes the undulant fever in human, and brucellosis in cow. Salmonella typhi, causal organism of typhoid. Shigella dysentery causes dysentery in infants. Corynebacterium diphtheria causes the diphtheria. Vibrio coma, which causes the cholera. The Salmonella paratyphi and Salmonella typhimurium, which causes the enteric fever. Streptococcus pyogenes variety scarlatti, which causes the scarlet fever. Aspergillus, Fusarium and Penicillium are able to produce different type of mycotoxins. So all these pathogens which are found in the milk which can resist the high temperature of the pasteurization are able pathogens. They can cause different type of diseases in human being. Now coming on to the different type of spoilage of the milk. Like we have studied the different type of spoilage of the meat, now we study the different type of spoilage in the milk. The abnormal fermentation of the milk is one of the spoilage. Why do we call it abnormal? Because we normally go in for the fermentation of the milk in controlled condition. As a result, we have the production of different milk products, the yogurt, the production of different type of sour creams, the cheese. All those are the process which refers to the controlled fermentation. But if you have uncontrolled fermentation, then it is called an abnormal fermentation, which is nothing but the spoilage of the milk, which can be caused by homofermentative Streptococcus, Lactobacillus, Pediococcus, which gives about 95% of lactic acid. However, there may be pathogenic heterofermenters like Mycobacterium, Micrococcus, which gives equal amount of lactic acid, carbon dioxide, ethanol and many volatile fatty acids. Gas production is another type of spoilage that is encountered in the milk. Now we have the production of carbon dioxide and hydrogen gas, which leads to the formation of a milk which gives a very bad taste and a bad smell also. This is called the foaming or the frothiness in sour cream and high fat milk caused by the species of Candida, Torula cremoris and many coliforms such as Escherichia coli, Enterobacter, Clostridium, etc. Ropey fermentation is another type of spoilage of the milk where we find that a viscous, slimy or ropiness structure of the milk appears. This is due to the production of the gummy polysaccharides or mucilaginous nitrogen structures being produced by different species of bacteria. We can have the microbial ropiness which is caused by Alkaligenus viscolactus and Enterobacter aeruginus. Or we can have non-microbial ropiness which is caused due to increased amount of WBC and fibrin protein which is coming out from the animal's body. Proteolysis refers to the degradation of the protein which is present in the milk caused by Pseudomonas, Bacillus, Proteus and Streptococcus liquefacients. Lipolysis, again like proteolysis, refers to the degradation of the lipid or the fat that is present in the milk caused by Pseudomonas, Achromobacter, Lipolyticum, Candida lipolytica and many different species of Penicillium. The bitty, or also called the broken cream and the milk, is another type of spoilage where a soft or a uniform texture of the milk or the cream is not seen. Sweet curdling of the milk occurs in the heated milk by thermophilic bacteria, which secretes the renin-like enzymes. Now this happens because we have the production of renin which coagulates the milk. This can be done by many species of Bacillus, Streptococcus liquefaciens, Escherichia coli and the species of Protease. The color change is again a very important type of spoilage of the milk. The different types of milk we can observe is the blue milk caused by species like Pseudomonas, Syncytia and Streptococcus lactis, yellow milk produced by Pseudomonas cinzanthus and Flavobacterium, red milk which is produced by Serratia marcensis and Brevibacterium erythrogenus, and we can have the production of the brown milk which is again a microbial fermentation because of the growth of all the mixed cultures of the spoilage bacteria. All these shows that the meat and the milk, which are very good source of protein, carbohydrate and water, is also a very good source for the growth of the microorganism. Though we consume it in order to have a good amount of protein in our body, we should be very careful that we are not consuming the spoiled meat or the spoiled milk nor the spoiled milk product, because the microorganisms may be present inside it and though it may not give any symptoms. So consume a good meat, a good food and therefore stay healthy. Thank you.